SN-38 bifunctional small molecules for localized estrogen receptor targeting. I'm Isabella.
And I'm Terry.
Yeah. Okay, so today we're going to first talk about some introduction to some modern anti-cancer drug delivery strategies, and then we're going to focus on RIPTACs, which is the focus for our presentation. Then we're going to talk about how that's related to hormone-dependent breast cancer and talk about our synthesis. Then we'll talk about a little bit of a side project that we've been working on and some future plans. So typically in drug discovery, what we've seen is a single function, like a ligand has a single function and it works on a single target. So, but what we've seen now is that we can tie two distinct drugs with two distinct functions together for a single compound with a hybridized function. Now, this can be important because this is important because it can allow us to enable more functionality, and it can allow us to access previously inaccessible spaces, and it can allow greater complexity in molecular function. So if you look at these three examples, the first one is a fluorescein tied with an RGD peptide. So the peptide localizes this whole molecule to cancer cells, while the fluorescein gives an optical readout. It's a dye, basically. So this is a molecule with two distinct functionalities. One is a dye and gives off the optical readout, while the other one can localize to the cancer cell. This next example is an antibody drug conjugate, and here we have deruxtecan, which is our toxic payload conjugated with an antibody. So the antibody localizes to a cancer cell, while our toxic payload can kill the cell. And this is an example of precision drugging, which allows less side effects and can be a solution to drug resistance. And the last example is a PROTAC, which comes from the Cruise lab. And in this example, we have an E3 ligand ligase, which is tied together with a ligand that localizes this compound. So again, two functionalities to treat cancer.
Yeah, and specifically our project focuses on a type of hybridized drug called regulated induced proximity targeting chimeras. And basically, this compound works through a ligand that localizes the entire compound to a specific cell that has a heightened expression of a target protein. For example, in cancer cells, this can be like a certain receptor, and that's conjugated with a linker onto an inhibitor that inhibits an effector protein that is essential for cell survival. And by optimizing the linker, this compound can basically induce cell death on the cells that it targets. And what's interesting about this is that the target proteins that the ligand binds to do not need to be the drivers of disease, and they also selectively target cancer cells. And this approach also has the benefit of being scalable compared to other approaches. And specifically for our project, we found precedence, especially for some of the components that we used in our compounds, from a chem-med-chem paper where researchers synthesized artemisinin and estrogen and artemisinin and tamoxifen to treat breast and prostate cancer. And they found that artemisinin estrogen had around an EC50 of 1 to 2 microliters when it was assayed against breast cancer cell lines. So we found that this was interesting because it could imply that this drug, as its hybridized function could be more potent than itself, standalone components. And yeah, specifically our project focuses on ER positive breast cancer, which is fairly common, and millions of women are diagnosed with it every year. And basically these cancer cells have a heightened expression of estrogen receptors on the cells or in the cell. And so our strategy when approaching this project is to use an estrogen receptor ligand to localize our compounds to these cells. Okay, and these are the different components of our SN-38 RIPTACs. Specifically for the estrogen receptor ligands, tamoxifen and raloxifene already have anti-cancer activity by themselves, but estradiol is just a steroid hormone. And for our first compound, we synthesized raloxifene SN-38, and this is our generation 1 synthesis. So basically, we first took the SN-38 and through monoacylation on the phenolic hydroxyl, we used TBS to protect that in order to ensure selectivity with our next step, which is acylating with an acid chloride to conjugate an alkyl bromide linker onto the hydroxyl. And then we conjugated that with our raloxifene onto the phenol, and then basically that yielded our final product. And this is the compound that we used in further cell assays, but the yield is pretty low, so we further proposed a future synthesis or future synthetic route that would help this problem.
Mm-hmm. And in this route, we start off with the silated SN-38 again, but this time we're using EDC coupling with 2-acetoacetic acid to form the AZ. And on the bottom, we have raloxifene, and we use 1-bromo-2-chloroethane in order to form this raloxifene compound. And then we can then conjugate those two compounds together to form a triazole linker. And we're still working on the synthesis, so we don't have yields yet. Okay, and then this was from the first compound that you saw. So this is our raloxifene SN-38 conjugate. The purple bars here on MCF7 are raloxifene SN-38. And we chose these two cell lines because MCF7 is an ER positive cell line, while MDA-MB468 is not. So what's important to note here is that we can see efficacy on from our compound on MCF7, but pretty low, like no efficacy on MDA-MB468. So we hypothesize this is because it is targeting and that our compound works.
Yeah, and our next compound that we propose synthesizing is tamoxifen SN-38, and we're approaching this through two strategies, basically installing the linker on different sites. So our first compound is O-linked to tamoxifen SN-38, and basically we started with 4,4-dihydroxybenzophenone, and then through McMurray coupling, we formed an alkene between propiophenone and our starting material to form bisphenol 4.2. And then we alkylated that to form the amine 4.3. And then we did, we conjugated a linker onto that. And then we did TFA deprotection. And then with 4.5, we can then conjugate that onto TBS SN-38 with an EDC coupling. And we haven't carried out we've only carried out the first two steps, so that's in future works.
And this is our N-linked synthesis. So the reason we're trying both N-linked and O-linked synthesis for tamoxifen is because we're not really sure how it fits in the binding pocket. So we're not really sure how conjugating SN-38 will affect its binding affinity. So we wanted to try both and just see how they would react in assays. So in the N-linked, we start off with just one phenyl, so 4-hydroxybenzophenone, and then we use nucleophilic substitution to alkylate the chloride on, and then we run McMurray coupling again with propiophenone, and then we can use we can substitute the amine, and we can use 16-anhydride ring opening chemistry catalyzed with DMAP to conjugate on our linker to tamoxifen. And then finally we're using EDC chemistry to EDC coupling to conjugate the tamoxifen and SN-38 together.
And this is our estradiol SN-38 synthesis. And this was our original synthetic route where we basically TBS protected estradiol at the phenolic hydroxyl to ensure selectivity. And then we did DMAP, we did a ring opening reaction with succinic anhydride to form an ester linker on the hydroxyl, but we were unable to conjugate this compound to TBS protected SN-38. So we went back to the drawing board and we came up with a new synthetic route that worked and also ensures or also shortens our synthesis. So basically through this, we first approached TBS protecting SN-38. And then with that TBS SN-38, we then performed the same ring opening reaction on succinic anhydride to form the ester linker at the hydroxyl on the lactone ring. And then with that, we proposed conjugating that with TBS estradiol and just using EDC coupling to form our final compound. And one thing to note here that is of interest is during the succinic anhydride or linker step, we originally had heated the reaction to 45 degrees Celsius, which we later determined to be too harsh of conditions for this reaction. But essentially what we observed is that the TBS protecting group fell off on the hydroxyl or on the phenolic hydroxyl. And this inspired further research on our part towards possibly using other sal ethers to protect that specific hydroxyl, which further kind of spiraled into a new project with SN-38s that will be covered later. And so for our future plans, we plan to experiment with linker length and polarity, as well as run more MTT assays on our compounds once we finish synthesizing them, and possibly also looking into Western blot and synthesizing toremifene SN-38 conjugates.
So now we'll transition into talking about our project with sal ethers on SN-38. Because our TBS protection fell off at harsher conditions, we synthesized two different compounds with bulkier sal ethers. And there's a precedent towards doing this with camptothecin analogs. So we have two here, DB67 and BNP1350. Both of them are on the C7 position though. Our sal ethers on C9 here. And what they found was that these compounds showed comparable efficacy in compared with camptothecin, but also stabilized the lactone ring here. So the issue with SN-38 in like the bloodstream is that the lactone ring like pops open or like hydrolyzes, and SN-38 is no longer effective. So they found that the sal ether helped to stabilize the lactone ring. Here are our three compounds and TBS here, TBDPS, and TIPS. You can see that the sal ethers are much bulkier on the bottom two. And on the right, we have our C-log-P and TPSA data, which is indicators of polarity.
Okay. We ran MTT assays on our C9 sal ethers to study their anti-cancer activity against colorectal cell lines as well as breast cancer cell lines, which we'll see later. But basically, we observed that SN-38 and camptothecin generally had or I'll explain the y-axis first. Okay, the y-axis on this graph is basically the cell viability, which is the percent of surviving cancer cells. And basically across our cell lines in general, you can see that SN-38 and camptothecin have the best activity. But in certain cell lines at certain time points, TB-DPS also has comparative, if not better activity than the original compounds, which is pretty interesting. So if you like go through the different slides, you can kind of see. Yeah, like these are colon cancer cell lines. This is breast cancer. Yeah, and we plan on doing topoisomerase assays to further study the relationship between the SN-38 binding to see, like, to further study the anti-cancer activity. So yeah.
Like before we move on, I just want to point out that TBDPS actually does show better efficacy than SN-38 and camptothecin at the 2 highest concentrations, which is pretty surprising given how bulky this sallow ether is. And this is a non-cancerous cell line.
Yeah, and this is just further computer modeling of C9 sallow ethers to kind of show how they bind to topoisomerase 1. And below that are the docking scores. And generally, like, the lower it is, the better. But yeah.
We also ran flow cytometry on our solid ethers, and the way it works is you stain it with PI and fitzinexin. Fitzinexin stains apoptotic cells. PI stains necrotic cells. And you can gate your cells based on the stain and form 4 different groups: viable, early apoptotic, necrotic, and apoptotic. And you can see our pie charts here. Like, to explain a bit further, we have PI stain on the x-axis and fitzinexin on the y-axis. So if the cell is in this upper right quadrant, it'll be apoptotic. If it's in this lower left quadrant, it's a viable, healthy cell. And we ran flow at 2 concentrations, 10 micromolar and 5 micromolar. And then, so for some future plans for our solid ethers, we want to try synthesizing these compounds onto an antibody as antibody drug conjugates. So right now we've tested conditions for the maleimide linker, which is the first step, and we found HCTU has the best conversion as a coupling agent. And then other stuff we want to run are BRDU assays and topoisomerase assays just to establish the functionality of these compounds. We want to thank ASDRP for this opportunity to do this research. All of our group members, our collaborator, Dr. John Wang, and our advisor, Dr. Edward New. Any questions?